consensus statement from a large group of scientists, and this arises from an effort that started in 1992, led by the Union of Concerned Scientists. And this was the first World Scientist Morning to Humanity, and included hundreds of scientists from around the world, including the majority of Nobel laureates in the sciences alive at the time. And there was the quite bold and, I would say, strongly worded statement that has not been the historical way that we talk about climate in the sciences, although it is increasingly becoming. The planet is not to be irretrievably mutilated. We really need to change how we steward the planet. And of course, all of you at the Jigecha Center there are intimately aware of this. In 1992, that was in 1992, and then 25 years later, this was followed up on relative to the benchmarks and guidance laid out in that first document. And other than working on the ozone hole, really very little progress had been made. And so they had a second warning. And my animations, of course, aren't coming up because I'm not in presenter mode so that it wouldn't be quite as large. So there is and arising from that, in order to make it as clear and transparent as possible to the global scientific and policy community, as well as the public, how those quite broad recommendations for climate stewardship and interventions should be applied, there has been a slew of discipline-specific present papers and consensus statements arising from this work, where primarily Bill Ripple has gone through and found experts in different fields, generated large teams within those fields to tackle the state of the science for climate interactions with these fields. And we're talking today about the first one, the scientist warning. Okay, we're going to skip over that. There is this quite nice behind the scenes, behind the paper article written by the first author of this, which was Riccardo Caviccioli, that I can recommend you to. So why microbes? Why was that one of the first discipline-specific papers that the group tackled? So many of you may already be aware of history. Sometimes they reveal entirely new metabolisms, or indeed have invented those metabolisms over the life history of the planet. So we're all familiar with the oxygenation of the atmosphere, thanks first to cyanobacteria and then eukaryotic algae and land plants, which steadily drove down the concentration of CO2 in our atmosphere and provided us with oxygen that was then able to fuel the metabolism of multicellular life. And so microbes, just as we see them interacting with CO2 and oxygen over here, they interact with a range of different components of the biosphere, and they really perform all the major metabolic pathways, and they periodically reveal entirely new ones. So some of the super cool things that have happened just in my lifetime of training include things like proteorhodopsin, so a light-driven proton pump that's present in between 40 and 70% of the cells in the surface ocean. And these are heterotrophic cells, which means that they consume other cells, other particulate carbon. They're not fixing their own carbon, but they're using sunlight as an energy assist. It's like if we were all walking around with solar panels strapped to ourselves that could allow us to go longer between eating. It's a fantastic adaptation in the low nutrient, low carbon surface waters of a great portion of the world's ocean. And that's just one example. We had no idea about that until around the year 2000. Okay, so another reason why microbes is that they contain a whole bunch of carbon, and that carbon is turning over quite quickly. So there's 10 to the 9th microbial cells per gram of surface water and 10 to the 6th cells on average per millet seawater. Between 50 and 90% of the marine biomass is microbial. And then here we have a visual representation of all the carbon in the biosphere of the planet, in the living organisms. And hopefully the glare is not too bad that you can actually, okay, so there's a bunch, and I'm just gonna use my hand because you can see my hand. So I'll point just like we were together in an actual room. Wouldn't that be nice? So we've got a bunch of carbon in the biosphere. Animals are down here, and people would be this little tiny square within there. And of course, there's a whole heap of carbon locked up in plants, but then all the rest of this are microorganisms. Okay, we've got archaea, viruses, bacteria, protists, which are just single-celled eukaryotes, and fungi. So collectively, that is a large amount of carbon, and it's turning over quickly. So unlike this carbon in plants and animals, which turns over on the scale of weeks, months, years, decades, and centuries for plants, these microorganism carbon forms are turning over really quickly. So when we think about a fast response of the biosphere to climate change, and sort of the quickest things that we will see in a lot of cases, barring things like fire and contagion, that's happening in the carbon stocks that are present in these microbial cells. Okay? And all of these little cells, all of the carbon that they represent, they're very busy doing things. And so they perform roughly half of planetary primary production. And as I understand from Dr. Matu, many of you are not microbiologists, and many of you might not be biologists at all. You're excellent climate scientists, glaciologists, modelers from a range of different disciplines. And so this fundamental fact about our Earth may come as a surprise to you, because remarkably, this is not something we teach children in school, and yet it seems profound enough that we probably should. Every second breath you take is thanks to marine microorganisms, so in terms of the oxygen we're breathing, and indeed half of the carbon that's being fixed on the planet that's associated with that oxygen being produced is from marine microorganisms. So we think about forests, but really half of the picture is these marine microorganisms. And this is this ecological champion, another organism that wasn't known until about 30 years ago, and yet is the numerically most abundant photosynthetic cell on the planet. And we didn't even know it existed until about 30 years ago. This is Prochlorococcus marinus. And here's just a tour of some of the beautiful phytoplankton that exist. These organisms, these microorganisms, are also master degraders. So without microbial recycling, nutrients would be locked up and not bioavailable to fuel further growth. So everything would grind to a halt if the nutrients stayed locked up in things. We need that composting, that recycling of nutrients to fuel further growth. Okay, and I don't know if you can see the bottom of my screen here. Move some of these things in case you can see those as well. All of this degradation is producing waste gases. And that brings us to our next point, that microorganisms are critical players in greenhouse gas cycling. So of the major greenhouse gases, with both natural and anthropogenic sources, microbes are the dominant mediators of their natural cycling versus things like geochemistry, so weathering, volcanism, etc. So we've got CO2, methane, and nitrous oxide. Okay, so CO2 is heavily microbially mediated. Microorganisms are responsible for roughly half of the consumption of CO2 and more than half of its production. Methane is fully microbially mediated, and N2O is mainly microbially mediated. Okay, so if we want to understand the trajectories of greenhouse gases in the atmosphere in the coming decades, we cannot ignore microorganisms, because they're responsible for a large amount of the natural background cycling that is happening behind the anthropogenic additions that we're making. Okay, they're indispensable, of course, to generating human food. So for our vegetables, there's crop microbe interactions and soil health that are essential to producing our vegetable crops. For our dairy products, There's rumen nutrition and fermentation. For breads, we have yeast, etc., and alcoholic beverages. So, and that's just to touch upon a few. So they're absolutely essential to producing our human food. They also produce a staggering diversity of natural products, including medicines, just naturally by themselves. And they can be engineered to produce synthetic products. So here's just an example of how bacteria can be genetically modified to produce human insulin. They also shape human health, and there's increasing indicators of how they can influence human personality and behavior. Things like whether you're irritable, how often you get hungry, your sense of satiety and well-being can be influenced by microorganisms. So there's roughly 10 times more microbial cells in your body than human cells, and 100 times more microbial DNA than human DNA. Humans are really superorganisms. And this leads me to a bigger point, which is all microorganisms are actually complex communities of microorganisms living within their hosts. And really, you can't understand host biology without considering the superorganism, which has a special name, which is Holobiont. You might hear some microbiologists or the media using this term Holobiont, and it just means the complete assemblage of organisms that is making up what we visually perceive as a single species of macroorganism. This really reflects the fact that a revolution is underway in how we understand biological life on the planet and human health. So I'm going to skip the next series of, well, this slide. This is in the paper, and you can see it in the paper. This is just a visual summary of some of the things that we've talked about. Okay, so we have the changing environment over here, and we have microorganisms here. So without explicit management, without us doing anything, without us being even aware of all of those interactions, the changing environment is impacting microorganisms through a range of mechanisms. Sometimes they will directly impact microorganisms. Sometimes they will change habitats that impact microorganisms. Sometimes they will change hosts that impact microorganisms. So this is already happening well underway. And without us doing anything, it will continue to do so. And that leads to things like changes in community composition, functional processes, physiological responses, and evolutionary adaptation in all those microorganisms, which, remember, are doing a huge number of services for humans. Now, this isn't just a one-way path, okay? Because of all those services that microorganisms do for humans, there are feedbacks between those organisms and our changing environments. And so these are changes that can, again, either directly or indirectly lead to exacerbation of climate change. If those microorganisms are making more greenhouse gases, then that will exacerbate climate change. And reciprocally, sometimes the microorganisms can be consuming the greenhouse gases and can be our partners, our allies, if you will, and can be consuming them. And without any understanding or management of this process, this is already happening, all of this. Now, with explicit management, we would like to push this part, right? We would really like to better engage our microbial allies in mitigating the full extent of climate change. We'd very much like to do that. What we would really not like to do is be managing for other things and miss the fact that microorganisms within one of our management strategies are actually totally counteracting whatever we're trying to do and are in fact making things worse, which right now, most of our management strategies, completely ignore microorganisms. And so very few of them are explicitly engaging them as allies, and almost all of them are totally ignoring whether this is happening at all. And so this may be happening as well, and it is. All right, so let's look. So basically, this is a big system of microbial levers that we can pull, either knowingly or unknowingly, and to interact with climate change. So let's look at some of those levers. For a series of global changes, we'll look at observed or most likely microbial outcomes. First, we'll look at a few examples in aquatic systems. So increased stratification, and for all of these, because you're a climate change center, I'm not going to discuss, as I would for a different audience, the details of these climate changes, because you all know a great deal about them, and many of you are experts in these. So for each climate change, we'll just take it as a given that yes, stratification is going to be increasing in many places. Okay, so for increased stratification in the tropics and mid-latitudes, where phytoplankton are nutrient limited, the decrease of mixing and increase of stratification decreases the nutrient supply. So we're getting less nutrients brought up from below. That decrease of nutrients decreases phytoplankton growth, that decreases the CO2 being fixed, and so that acts as a positive feedback to increasing climate change. And remember, because phytoplankton are responsible for half of global primary production, changing how their dynamics are working and how much carbon they're fixing can really have a big impact. Okay. However, in the high latitudes, okay, this is our first indication that for each of these responses, things can vary a lot based on where you are on the planet and what the specific details of the situation are. So in the high latitudes, you don't tend to be nutrient limited, you're light limited. Okay, so here you've got plenty of nutrients there, and increased stratification allows the phytoplankton to stay in the surface lit waters for longer. So here, this leads to increased time in the photic zone, increased growth, increased CO2 fixation, and a negative feedback to climate change. Okay, so another example from aquatic systems is increased nutrient input. And many of you will be familiar with this because it's in the popular press so much, the idea of eutrophication. Water bodies, we can have sewage, etc. And all of these things are perturbing microbial ecology. And higher coastal nutrient runoff can occur due to heavier rains. So the more extreme precipitation events we get are a synergistic interaction, have a synergistic interaction with eutrophication in general. Okay? So what does that do to the microorganisms? Well, first of all, increasing nutrients alters the microbial communities, for sure. There's lots of evidence of that. And one specific thing that we might really care about is that increasing nutrients often leads to harmful algal blooms, HABs. Okay, stratification can also contribute to HABs. And so these species that are involved in harmful algal blooms can be more competitive under stratified conditions, at warmer temperatures, and at the resulting lower viscosities. So we have both eutrophication and stratification leading to blooms of these harmful algae. And as many of us know from either direct experience or reading about this in the press, this can lead to fish die-off, health impacts as some of the chemical compounds released by these harmful algal blooms go into the air and make us and our loved ones sick, etc. Okay, and it can turn water bodies entirely thick with soup, basically. Okay, so another key driver is ocean acidification. So the pH has already declined 30% in the last 250 years, because this is it's a log scale, and so a small difference that we've seen is actually reflective of a 30% decrease in pH, and it continues to drop. So acidification makes it harder for calcium carbonate to form, and a lot of organisms have shells or skeletons made of calcium carbonate, and there's a lot of macroorganisms that we care about. You've likely heard about corals as an example of this. There's beautiful things like this mollusk, it's called a sea angel. It's very beautiful, Cleone limacina. Echinoderms like sea stars, crabs, lobster, etc. And this impacts the entire microbial food chain. Let's look specifically about some microorganisms rather than just the charismatic large organisms. So here's a great example, coccolithophores. These have calcium carbonate coverings. You can see these plates here. Their name is for round stone bearer, and those little stones are coccoliths, and they cover the surface of these organisms to protect them. They form blooms in the Sargasso Sea, in the Gulf of Alaska, and a number of different places around the world, and then they settle into chalk layers, and so you can actually see them in the geological record around the world. They formed, for example, the White Cliffs of Dover. Okay, another example is foraminifera. These are all over the place. So here, they've got these beautiful shells. This is an example here. And so these calcium carbonate shells are perforated, and there's chambers inside. They're mostly heterotrophic. These guys are phototrophic. These are heterotrophic. They eat bacteria, phytoplankton, and small plankton. They're really abundant from 40 north to 40 south. And so these are just two different types of microorganisms in the ocean that have consequential ecological roles, are abundant, and are already being impacted by the drop in pH, and are going to continue to be even more egregiously impacted. So that's just two major groups. There's a strong expectation that acidification will impact communities overall, not just species with calcium carbonate shells, and not just indirectly by these organisms dropping out over time, since pH is known to be a major driver of microbial community composition in terrestrial systems. So there's also, we're going to skip the nitrogen example since we're short on time now, but suffice it to say, nitrogen deposition impacts these organisms as well. And there's a host of other things like oxygen minimum zones that all play into how aquatic systems are changing. I'd like to give one brief example, since so many of you are specialists in glaciology. This is from a postdoc. This is a closer look here. This is from a postdoc who is co-advised by myself, Dr. Zhiping Zhang, and Lonnie Thompson and Ellen Mosley Thompson, whose names many of you will be familiar of because they're wonderful leaders in glaciology and just great human beings. Many of you may know them as personally because they've been active in the community for so many years. And so Dr. Zhang is co-advised by them and Matt Sullivan, who's a virologist, and myself here at Ohio State University. And so one of the things that Zhiping has been looking at is how glaciers can reveal how paleoclimate and ancient microbiomes interacted in the past. So of course, these microorganism climate change interactions aren't just happening now, they've happened through the extent of human history. And what can the record in our quickly vanishing glaciers tell us about those interactions? Okay, and so you may be aware of, but if you're not, this may be of particular interest to those of you who do glaciology work. Thanks to Ellen and Lonnie Thompson, there's this incredible global ice core archive at the Burn Center here at OSU. Here's just a map representation of all the different places that Ellen and Lonnie have brought back cores from. And so there's more than 7,000 meters of ice stored here. And here's Ellen and Lonnie Thompson inside the archives. Okay, and specifically, Zhiping was looking at cores from the Goulia core in the third pole. Okay. Now we're just going to skip straight to some of the results. Each of these dots here represents a different microbial community in an ordination such that dots that are closer together are more similar, and dots that are farther apart represent less similar communities. And so Zhiping broke this into high and low dust sections of the glacial core, into cold and warm temperatures, and to climate epochs of the Holocene, the last glacial period, and the pre-LGS, pre-last glacial. Okay, and what you can see if you start squinting and looking at this, which we don't have quite enough time to go through, is that the communities differ by climate epochs statistically significantly, and they also differ by temperature statistically significantly, but not by dust concentrations. And that's interesting, and we got quite a bit into some of the reasons behind this and what we see. And this extends not just from whole communities, but to specific strains of specific lineages. So this is just one example where each row represents a different strain of this particular Cryobacterium genus, and each column represents a depth in the ice core, from most recent, most shallow in the core to oldest in the core. Okay, and this just shows you the presence and relative abundance by the intensity of each of these cells of these different organisms through the core. And you can see that there's really quite a big difference as we go from more recent times to old times in the dominant lineages of these organisms. There we go. And this can actually be linked directly to single nucleotide differences among these lineages. So if we think about, we can actually start to get to some of evolutionary studies, which Shiping is underway right now, where he's recovered entire genomes from these organisms from the ice. And so he can see how these particular sequence 2 and sequence 3 that were the dominant types actually perhaps evolved over time. He's doing the reconstructions right now, evolved into this dominant sequence 1 that we see here. So there's higher microbial concentrations and diversity in cold periods. And this isn't just driven by differences in dust. Okay, again, it's not just a dust impact, because dust is higher during cold periods a lot of times, but this is independent of that. Okay, the most parsimonious explanation that we all think of when we think about, cold periods perhaps having higher diversity is that, well, the winds are stronger and the capture area is larger for these microorganisms. But in fact, in this particular area, in the high Himalayas and this third pole region, this is part of the Tibetan Plateau, and they've done current day reconstructions of the capture areas and capture extent of different wind conditions, the monsoon, different source conditions, etc. And it really does not seem to be supported by a larger distance or a wider area, because everything, like there's a huge area up there that is all mountainous and arid. Okay? But what does seem to be the case is that cold periods had longer snow at the top, longer time where the snow, the fern at the top of the glacier, stays there before it gets compacted. And that was providing a longer time for microbial growth. And so there's a suite of analyses that Dr. Zhang has done that really support this, that show that there are distinct communities with different phototrophic foundational members, bases of the food chain, and entirely different food chains, representing short fern periods. Again, fern is the top. For those of you who are not glaciologists, which is probably a chunk of the audience as well, the fern is the kind of uncompacted snow and partially compacted snow at the top of the glacier before it turns all the way into ice. Okay, and the microorganisms are living there. So those microorganisms are both those that have gotten deposited from all over the place from the wind, and those that are thriving there and replicating there before they get buried, basically. So a number of analyses that we don't have time to talk about today really support that there is this connection between climate and the conditions at the tops of glaciers that can be seen in the signatures of those communities in ways that we're just beginning to look at from their entire genomes to now start to ask, were there differences in their metabolisms under these different conditions? Okay, Jiping also collected viruses from these samples, and the goal was not to look for pathogenic viruses. This is a very remote glacier. Excuse me. The goal was to look for viruses that were infecting microorganisms. And so here he specifically looked at two particular depths because there was a lot of additional methodology and deep sequencing that was required to be able to recover the viral signal here. We're not talking about looking at the viruses visually, we're talking about looking at their DNA sequences. Okay, so for these depths, he compared the viral communities and the microbial communities from those two depths to try to understand what the viruses might have been doing ecologically in terms of those microbial communities. And so again, we have this what's called a heat map, where each cell's intense color intensity denotes the abundance of a particular group. The different groups of viruses now in this case are on the rows here, and the two columns are just those two different samples. Okay, so the majority of these viruses we can't really say anything about because they're unclassified. And this is really common in environmental virus studies, where they're just things that nobody's ever seen before. We don't know anything about them. But a subset of these organisms were able to be identified. Okay. And they can be linked to microorganisms that lived there as well. Okay. Specifically, there were several viral types that were inferred through reconstruction of their infectious profiles to infect Methylobacterium, which is interesting, of course, because we use methane concentrations in bubbles in glaciers to inform us about historical climate. And if there's evidence for methanogens there, and even the viruses that infect methanogens or methane consumers there, then that can really be altering the concentrations in those gas bubbles of methane after deposition in ways that could decrease the strength of our inferences about ancient climate. Now, this is not saying that those things are in question yet. This is just starting to see some things that make us go, huh, what does that mean? So we don't know enough to say more yet about whether this tells us anything about the actual gas concentrations and whether they were altered. We can just see that there's organisms that could have altered them that were present. Okay, so we are just about out of time. So I'm just going to say that in soils, a bunch of things are going on as well. As things get warmer, organic matter degrades faster. So all of those little microbial cells, their enzymes can work faster, and organic matter degradation increases. The presence of organisms changes, which organisms can survive change, and this is due to both changes in temperature and in precipitation and changes in soil type. We don't really understand, people, this is a very active area of research. Because microorganisms grow and divide and die so quickly, they can be evolutionarily adapting to these changing conditions in ways that could really change how they're responding to the various components of climate change. But this has not been sufficiently characterized in different study systems for us to understand which way this is going to go. So when we think about those levers and the dangers of pressing those levers without realizing that we're pressing them, this is a whole other area of like, those organisms are changing right now and changing rapidly over the next decade and 2 decades in ways that even if we understand what they're doing right this moment, which we don't, it's very difficult right now to understand how they're going to change in their abilities in the next couple of decades. So there's a lot of active and important research happening in this area. Okay, so there is a great paper that summarizes some of these things that is here, and I'll get the sharing permission cleared up with Dr. Matu, and so that she can actually share this whole PowerPoint with all of you in case you're interested in looking at it later, and in case you're interested in looking at that particular paper. Okay, so I just like to mention methane is really important too, where you have things getting wetter. Methane is a big deal. Land use change is interacting with microorganisms. Okay, so land use change absolutely impacts soil microbiota. So if we go in and we start managing a system to be a grassland for farming, or convert land into an agricultural crop area, the microorganisms will end up being very different between those things. And this impacts drought resilience and climate change resilience more generally. So in general, grassland's resistance to drought conditions is higher than cropland's resistance, partially because of what's going on under the hood of these systems in terms of the microorganism community diversity that's there. And there's this lovely closer look from a member of my team that we're not going to have a chance to tell you about, but in the Sahel, my student is looking, co-advised by another faculty, at the impact of co-cropping between a shrub and millet. And this is like, ignore all the rest of this since we don't have time, and just look at the difference here. So here, this is the plush shrub. And shrub intercropping is a traditional practice there that's been around for a long time. This isn't anything new. But when shrub intercropping is allowed to occur, the yields from the millet crop are way better, and the drought resilience is way better. So here is minus shrub, here is plus shrub. Very good visual difference. Okay? And so she is investigating the microorganism relationships between the shrub and the soil and the millet that may be contributing to this increased drought resilience. I'd like to just firmly, in fact, and the most important thing here is to go back and acknowledge the other people who made this work happen, which is, so Laura Mason is the PhD student who's driving it, and this represents work that's been done over several decades by Professor Richard Dick, who's in another department here at Ohio State University, and a suite of people from the two labs have been contributing to this. But this really arises from this long-term work that Professor Dick has been doing in the Sahel. Okay. Oops. Okay. So we're going to skip why it's very important, because I'm guessing that as a climate change group, you understand that it's important, and we saw this. So now let's look at this one a little bit more. So by having this shrub intercropping in the fields of millet, even without any fertilizer addition at all, you already get a very strong increase in millet yield. Okay? And if you add some fertilizer, it's even better. But even without any fertilizer, you get a very strong yield increase. The yields are maintained in low rainfall years, and this increases the water use efficiency. It appears that the shrubs are performing hydraulic lift, which means they're bringing up water from deeper and bio-irrigating the nearby surface soils in a way that is directly benefiting the millet during times of drought. But this hydraulic lift really isn't producing much water. It's really quite a small amount of water that's being provided to the millet. And so physiologically, this cannot be the whole story for how much better the yields are and how much better the drought resilience is. And other studies, but primarily Laura's work, we believe that the beneficial microbiome from these Gera shrubs is part of what's contributing to that millet resilience. So how does that work? So over here, here's a millet plant all by its lonesome, and it has a, one type of microbial community. The millet plant that lives next to a shrub, where periodically every year the farmers cut off the above ground material from the shrub and just let it sit on top of the ground, or sometimes they burn it and mix it in. This millet plant is experiencing a quite different microorganism below ground, and this is established. Laura sees this consistently in a range of locations in the country, a range of different conditions. Excuse me, that the microorganisms are different. So there is some zone of influence around the shrub that is impacting the millet. The microorganisms are different. There's this hydraulic lift that's happening here. That could be one of the ways the microorganisms are different. There's increased moisture thanks to that hydraulic lift. There's also increased carbon from the carbon inputs from the shrub, and this increased plant biomass in the overall system. So the microbial community can support plants through drought in a couple of different ways. These are color-coded green for direct benefits to the plant, and brown for indirect benefits through soil changes in soil properties. So this is overall microbiome support for the plants. So we've got plant and microbial osmolytes. So the microbes can actually change the plant osmolytes by producing hormones that cause the plant to behave differently and to produce its own osmolytes differently. And the microbial osmolytes in the soil can change the overall soil property. They are contributing to increased soil carbon, which can increase soil health and moisture retention capacity. There's increased nutrient availability from these microorganisms that interactions of the plant, and antioxidant production, which can decrease the stress of the plant. So right now, Laura's in the process of looking at the collection of genes from the entire community, which we call a metagenome. So meta for the entire community and genome for all the different pieces of the genome. She's looking at metagenomes from these conditions of the soil and right around the shrub and the millet rhizosphere, and seeing where she sees changes in the genes that encode these different functions. So right now, some of these have been observed, some of them are hypotheses, and she's going through and looking for the individual genes responsible for each of these things. Okay. So methane's a big deal. Dr. Matu, I think that I'm still going to need to skip a lot of that, but thank you for letting me talk a little bit about Laura's work. I appreciate it. So wastewater treatment in ruminants, constructed wetlands and rice paddies all produce important amounts of methane. These human sources are increasing as humans expand our footprint on the planet and change our land use management practices. One thing that's near and dear to my heart, and that the institute I co-direct specializes in looking at, is these thawing permafrost systems. And here's just an example. This is a boardwalk that we use to walk through the systems to try to disturb them as minimally as possible. And you can see that just over a couple of years, this boardwalk has now sunk into the ground. And that's because as the permafrost thaws, the entire ground slumps. And depending on the local hydrology, it can just slump and stay dry, or it can slump and get wet. This ancient carbon, there's twice as much carbon locked up in the permafrost as there is in the atmosphere, which is extraordinary. And this ancient carbon that has been stuck in the freezer for thousands of years, in the sort of planetary freezer, the freezer's now failing and that carbon's becoming bioavailable. What happens to that carbon is really consequential because there's so much of it. If it stays put, no problem. If it comes out, that is a appreciable feedback to climate change. And it matters whether it comes out as CO2 or as methane because of the much more potent global warming potential of methane. Whether it comes out as CO2 or methane depends on the hydrology and also on the microbial dynamics. So as the community thaws, you've got changes to the microbial community. Okay? And you see, anywhere you've got wetness, you see an increase in methanogens, so organisms that make methane. You also see the presence of methanotrophs, organisms that consume methane. So those would be an example of our allies, if we go back to that first conceptual slide. So can we think about how to manage systems to partner with our allies, the methanotrophs that already live there, so that they can more effectively mitigate the release of all of this carbon as methane. Even if we have it come out more as CO2, even if we can't help that it's going to come out maybe, but if it comes out as CO2, that's going to do a lot less damage than if it's coming out as methane. And the methanotrophs are chewing this, using this methane and converting it to CO2. Meanwhile, plants are expanding in these areas as the ground is thawing. Root exudates are therefore increasing. That's fueling microbial activity, and that's allowing them the priming of old carbon that was really, or even more recent carbon that was really recalcitrant and hard to break down. with these added root exudates from plants, that's like juicy, good carbon. So it's like putting a coating of sugar on top of broccoli, right? It's going to make it go down a lot more easily for, just to use a funny example, make it go down more easily for the microorganisms. Okay. However, the plant growth itself is sucking up CO2, and so that's increasing CO2, decreasing CO2. So this just highlights yet another case where we have a complex interplay. There's a bunch of things about the human health, and human health is also greatly involved with pathogens. Okay. So, a lot of this work reflects this cartoon, which is that the research is concluding we're destroying the earth, and the policymakers and government are saying, well, what do you really mean by that? And so this is something that's posted on the Scientist Warning website to sort of capture the fact that we need to be as explicit as possible in as many ways as possible, not just say, hey, there's a problem, but go into each of our separate disciplines and say, here's how climate change is impacting our discipline, and here's what we can do about it. And so the sort of final part of this paper is this box 2, which is a call to action. And a lot of these things are, quite intuitive. They're things that seem quite reasonable. And yet this is putting a very explicit point on the importance of these things. So a greater recognition that all multicellular life relies on microbial life. A number of different research and development priorities, that microbes be included in basic climate change research, that experimental design includes monitoring of microbial response and feedbacks to climate change, longer-term monitoring of those feedback mechanisms, incorporation of microbial processes into models, and I think the engineers among you will appreciate this, the development of innovative microbial technologies to minimize and mitigate climate change impacts. In teaching, really including this microbial interconnectedness in diverse curricula from K through 12th and beyond to appropriately train scientists, engineers, and the workforce in the fact that there are all these levers happening, whether we know about them or not. And so we really need to know about them and understand them so we can press them correctly as we manage the climate system and the planet system. And policy, trying to include explicit consideration of microorganisms in policy and management decisions. And that includes their incorporation in thinking about the UN Sustainable Development Goals. Okay, so that pretty much concludes what I have the opportunity to tell you about today, and I do apologize. I'd just like to flag that we have several training opportunities associated with our institute, which you can find at this website and on Twitter. And so we have an undergraduate summer research opportunity that happens in Arctic Sweden and in New Hampshire in the United States, and that's happening every year. We have a professional summer program. This just says June 2023, but it's going to happen, I think, in 24 as well. We just have been having it this summer so far. It's been going great. So this is for people who already have a bachelor's degree. There's a new PhD training cohort program with PhD cohorts entering this year, next year, the year after that, and the year after that. And with that, and so I didn't actually get a chance to tell you about the Institute today because the focus was more broadly on how microorganisms interact with climate change. But in terms of our Institute, we're trying to identify the rules by which microorganisms and ecosystems generally exhibit emergent responses to climate change. And we have over a decade of high quality multidisciplinary data from a Thawing Permafrost site, and we are always excited to engage new collaborators because there are way too many questions for us to be able to answer all of them. And there's lots of great data that's already in hand. And so we're delighted to forge new collaborations.
Thank you, Virginia, for this excellent talk, which has really put humanity on a notice. And we really need to get on to doing a lot more research on microorganisms and climate change. I now leave the floor open to everybody. Whoever wants to ask a question, please raise your hands and mute.
Can I ask a question?
Yes, ma'am.
I was just curious to know, you mentioned that the glaciology microbe studies are done from using ice cores. Are these from the ones that are stored in your archives?
Yes, that's right. So we put in a proposal recently to partner with another organization as well to look at some of their ice cores. But so far, all the work that we've been doing over the last 5 years that's been led by Dr. Zhang has focused on the Galea ice core from our archive. And the first step was working out a rigorous methodology that avoided contamination while the ice core was processed, because as you might imagine, the biomass of the microbes and viruses inside that ice core is pretty low. And so the opportunity for contamination while processing is high. So he spent a year and a half developing rigorous methods for, I wouldn't say avoiding contamination because you can't entirely, but minimizing contamination and then characterizing it so that any signal from contamination can be subtracted from the signal of what's actually in the ice core.
Thank you. And I also have another small question. Have you found the viruses to be the same in the yesteryears as well as now?
Great question. No, there's actually a lot of change over time. There are some viruses that are persistent. So both for viruses and microbes, Dr. Zhang has identified and described cohorts of microbes and viruses that are historically persistent over the roughly 500,000 years spanned by this core and his samples from it. And I say roughly because, as many of you know as glaciologists yourself, the dating process is rigorous and takes a long time. And so they're thinking that it's actually probably like 650,000 years old at the bottom of this core, but they're still in the process of doing all of the demonstrations of that. So regardless, it's a long span of time, and there is a group of microorganisms that are stable over that time, and then there's a lot of other ones of both microbes and viruses that are changing.
Thank you, and it was an amazing talk. I would have wanted you to go on because it was really interesting and very, very informative. Thank you so much for this wonderful talk.
Thank you, and I know it's the end of your days there, and I appreciate your staying, and I'm just so sorry about the technical issues.
Our pleasure to hear you.
Dr. Virginia, let me congratulate you. Wonderful talk. We missed the big screen and also the health-related issue on human. That also we'd like to hear one more time. One thing I have, you are talking about the permafrost, doing that one. Nearly 33 new germs have been identified, and most of them might lead to pandemics in future, like COVID, what we had. Do you anticipate these pandemics in future?
That's a great question. I think it is worth our attention and our concern. It is hard to, we know the carbon is now bioavailable, and we know that some of it is coming out as CO2 and some of it's coming out as methane. So that is a known danger. The potential for ancient pathogens to reemerge from the permafrost, or indeed from glaciers that are melting rapidly and coming down into our villages and towns, that is a potential danger, and the carbon is a known danger. So I think monitoring and careful tracking of that is important. And it's definitely worthy of concern. I wouldn't want to speculate on whether we're going to see a new pandemic coming from the permafrost. So far, we've seen, you've probably seen, there's been, you know, an outbreak of bubonic plague at one point in Alaska that came from a graveyard after the permafrost had thawed, and some things like that have come out, but those haven't spread and turned into epidemics or pandemics yet. I do think it's absolutely, and I know that like the US military is putting funding into observing that. I have colleagues who are microbiologists with the US military, and they're funded to study pathogens in the permafrost for exactly that reason.
Thank you very much.
Thank you so much.
Wonderful talk. God bless you. We like to hear a lot more.
Thank you so much, Dr. Permafrost. I appreciate it.
Thank you.
Do we have any more questions? So if you don't have any more questions, let us all thank Dr. Virginia for this excellent informative talk, where a statement also which had been previously released as a scientist's warning to humanity. And I think we've got a direct first-hand experience in hearing from you. So thank you, and we hope we can invite you again so that we can discuss in a more elaborate manner about your work. And in fact, the work on millets has come as a big surprise to me, because I did not know that aspect of your work. And we're also at the Nature Center, we're also involved in work on our work involving microorganisms and millets. So thank you once again for this wonderful talk. And I just request some of